Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A Level Biology Unit 5 for January 2021. This is the part 2 video. I'll put the link to the part 1 video below the description box. Let's begin with question 5. Question 5 says, one cause of blindness is a mutation in the DNA coding for the enzyme RPE65. The enzyme RPE65 converts trans retinol to cis retinol. Explain why mutations in the RPE65 gene can result in blindness. According to this question, this enzyme is responsible for the conversion of trans retinol to cis retinol. And if you can look at my summary down here, we know that inside rhodopsin we have retinol in the cis form. However, when light strikes it, the cis retinol is converted into trans retinol. And this trans retinol breaks down into opsin and retinol. These are recombined to be able to produce the cis retinol. So it means that enzyme participates in this reconversion to produce rhodopsin in the cis retinol form. So it means if the enzyme is not working well, then this process is going to be hindered, so we cannot regenerate the light-sensitive pigment inside the cells of the eye. So here I said the mutation leads to formation of an enzyme whose active sites are not complementary to the transretinol. Remember, transretinol will be the substrate for this enzyme. So cisretinol cannot be regenerated from transretinol and rhodopsin cannot be reformed, so the rod cells will lack the photoreceptor. And again, remember the photoreceptor is the rhodopsin, which is present in the cis form. When light strikes the rhodopsin, is converted into the transretinol form, which breaks down into opsin and retinol. These are later recombined with the help of the enzyme to produce the photosensitive pigment, which is rhodopsin. Let us continue to the next part. Part B says, scientists are investigating the use of gene therapy to treat blindness caused by mutations in the RPE65 gene. In one investigation, different gene therapy doses containing functioning RPE65 genes were compared. 2 months after treatment, the transretinal and cisretinal in each eye and the ability to follow a path were measured. The graphs show the results of this investigation. This first graph we can see the vertical axis is showing us the quantity of the transretinol, and the horizontal axis is showing the gene therapy dose in arbitrary units. So we can see as the dosage increases, the quantity of transretinol is varying, and we can see that data has error bars which show the variation for each dose. The second graph shows the quantity of cisretinol, and overall we can see it decreases and then it increases as the concentration of that dosage is increased. So there will be more cisretinol. That is because it has to be regenerated. The third graph shows the ability to follow a path correctly. Here we can see that ability overall is being increased with the increase in gene therapy dose. Let's continue to the next page. Here they ask, describe the conclusions that can be made from this investigation. They want you to use the information in the graphs to support your answer. Looking back to the graphs, I came up with this conclusion. I observed that the higher the gene therapy dose, the more cisretinol produced in the eye. Also, from the data we can see as the dosage was increased, cisretinol must have been formed. Therefore, the higher the gene dosage, the more the conversion of transretinol into cisretinol. Also, the higher the gene therapy dosage, the greater the ability to follow a path. And finally, I said, vision is therefore restored as more cisretinol is formed. When we look at the data sets given, there were error bars that were put. However, some of them did overlap. So based on the error bars, we could say that data may not be that dependable or may not be that valid due to the overlaps observed in the error bars. So let's continue to the next page. Here they say mutations in other genes can result in blindness. Describe how these genes could be identified. Imagine we did not know the genes that lead to blindness. How can we be able to identify them? All we need to do is we have to use microarray technique to identify any genes that are actively transcribed into messenger RNA in the eye. And then we will sequence the genome of people with inherited forms of blindness. And we'll compare the sequences of active genes from large numbers of people. These are the people with the blindness and those without blindness, basically, to identify the mutations causing the blindness. This brings us to the end of question 5. Let's continue to question 6. Question 6. Exercise affects the cardiac output of a person. The graph shows the change in heart rate following exercise for an unfit person and for a fit person. So let's look at this graph. I put some values based on how I answered the questions below. So the vertical axis is showing us the heart rate in beats per minute, and the horizontal axis is showing us the time after exercise. Our general observation is the people who are unfit have a higher heart rate because after exercise we can see the heart rate continuously remained high for the unfit person in comparison to the person who was fit, and there was a major drop for the person who was fit in comparison to the one who was unfit. So here they say the heart rate of the unfit person decreased by 16%, 120 seconds after exercise. They want you to calculate the difference in percentage decrease in heart rate of these two people, 120 seconds after exercise. And they want you to give your answer to an appropriate number of decimal places. So I began by using this value here that was given. So the percentage decrease of the unfit person was given in the question. So it was 16%. I also went to say the percentage decrease of the fit person. To calculate the percentage decrease of the fit person, I got 170, which is this data here, minus the 90 at 120 seconds, then divide by 170, which is the starting value, times 100, and it gave me 47.06%. So since I have the two percentages, getting their difference was easy. So 47.06 minus 16 gave me 31.06, which is roughly 31%, and this was my answer. I did a side part here to show you how the 16% could have been calculated, and this is how you'd have got the 16%, although it was given to us in the question. So let's continue down here. They say calculate the heart rate of the unfit person at 5 minutes. The change in the heart rate of the unfit person is a linear relationship. Use y is equal to mx plus c. Now, because the question stated that this is a linear relationship, I'm going to assume that it's a straight line, and therefore the gradient of the whole straight line is always the same no matter at what time, whether it's at time is equal to 0 or time is equal to 240 or time is equal to 5 minutes, it's going to be exactly the same. And again, the time here is seconds, so if it's 0 seconds, 240 seconds, or 5 minutes, it doesn't matter. Since it's a straight line, the gradient is going to be the same. So I used the data provided here, and I did gradient of a line is going to be change in y, which is 190 minus 125, divided by change in x, which is 240 minus 0. So here I got, because this is a negative gradient, of course, 190 minus 125, it gave me negative 65, and the change in x is 240, so the gradient should be negative 65 over 240, which is negative 13 over 48. Since I know this is the equation of a line, whereby m is the gradient, x is the x value, and c is the y-intercept, I know this should touch the y-axis at the y-intercept, which is 190. So substituting 190 for c, we know the gradient is negative 13 over 48, and the x-coordinate, remember x is going to be time. However, they've given us 5 minutes, so I had to convert this to seconds. So my final answer was that times that plus that, which gave me 108.75, which is rounded up to about 109 beats per minute. That is my answer. Moving on. Here they say explain the difference in the time it would take for these two individuals to recover from exercise. During exercise, there is going to be a lot of activity and some anaerobic respiration is going to take place, so there will be some oxygen dead, and after exercise, that oxygen has to be taken into the body. However, because a person who is unfit does not constantly exercise, their lung capacity is going to be low, their rate of gaseous exchange is going to be lower in comparison to the fit person. So, I said, due to constant activity, the fit person has greater cardiac output, and the fit person has greater lung capacity. The fit person has better gaseous exchange. The fit person can easily overcome oxygen debt for the heart rate to rapidly normalize after exercise. I put something here in red. You're not supposed to put it there if you don't want, but just as a reminder, I said constant activity makes the heart and lungs work or train. It also allows the muscles to deal with lactic acid by eliminating it faster. It also allows the liver to rapidly oxidize lactic acid once oxygen is sufficient. The next part says describe how heart rate is controlled in response to changes in the blood following exercise. I say during exercise, lactic acid and carbon dioxide production cause a decrease in blood pH. The chemoreceptors in the blood vessels send impulses to the cardiovascular control center in the medulla, and a response from the medulla through the sympathetic nerve causes the sinoatrial node, SAN, to increase stimulation of the heart muscle. Once the blood pH returns to normal, or it increases in this case, this change is detected and the medulla sends impulses via the parasympathetic nerve, and this parasympathetic nerve causes the sinoatrial node to slow down and the heart rate is going to be normalized. So this brings us to the end of question 6. Let's continue to question 7. Question 7. Parkinson's disease is a nerve system disorder that affects movement. In people with Parkinson's disease, some neurons in the brain die. Many of the symptoms of Parkinson's disease are due to a loss of neurons that produce a neurotransmitter called dopamine. Describe how a neurotransmitter transmits a nerve impulse across a synapse. I said, impulses arrive at the presynaptic neuron, and this causes the voltage-gated calcium ion channels to open. The calcium ions diffuse from the synaptic cleft into the presynaptic neuron, and here they fuse with vesicles containing the neurotransmitter. Then the neurotransmitter is released into the synaptic cleft. It will diffuse and then bind onto the receptors on the postsynaptic neuron. When that occurs, the ligand-gated sodium ion channels will open. Then the membrane of the postsynaptic neuron will be depolarized. If a threshold is reached, then an action potential can be generated in the postsynaptic neuron. This is very easy to remember and is exactly as it is in the textbook. So let's continue to the next part. Here they say, one treatment for Parkinson's disease is a drug called L-Dopa. Describe how L-Dopa reduces some of the symptoms of Parkinson's disease. L-Dopa can cross the blood-brain barrier, and also this L-Dopa can be used as a precursor for production of dopamine, so it's going to be very helpful in the treatment of Parkinson's. So I said, Parkinson's disease is caused due to the death of dopamine-producing cells in the motor cortex. Symptoms include muscle tremors. This is one symptom of Parkinson's disease. Also, L-Dopa crosses the blood-brain barrier. The body can use L-Dopa as a precursor to make dopamine, and this replaces the lost dopamine and restores normal muscle movement. Let's continue down here. Here they say scientists can induce pluripotent stem cells to produce transcription factors involved in the synthesis of dopamine. Explain how transcription factors are involved in the synthesis of dopamine. If you remember, transcription factors will bind to specific genes that are to be transcribed. So here I said that transcription factors bind onto the genes involved in the production of dopamine, and this enables the genes to be transcribed into messenger RNA. The messenger RNA is used in translation to synthesize dopamine. This is a 2-mark question, so it's simple like this would earn you the marks you're looking for. Moving on. Here they say some people with Parkinson's disease have a DNA mutation called LRRK2. Scientists studied the effect of this mutation on the dendrites of dopamine neurons produced from the stem cells of 3 groups of people. The graph shows some of the results of their study. So here we can see from the first graph the mean number of dendrites per neuron. People without Parkinson's disease, of course, have more numbers of dendrites, while people with unknown causes of Parkinson's disease and people with Parkinson's disease caused by LRRK2, their number is almost the same. If we go to the second graph, we see the mean length of dendrites. People without Parkinson's disease have longer dendrites in comparison to those with Parkinson's disease. However, we can see people with Parkinson's disease caused by LRRK2 have greater length in comparison to those that have unknown causes of Parkinson's disease. However, we can see the error bars do overlap, so that could show that data may not be that valid. There is no overlap with this data, so we can see this one is valid, but these two overlap, and that could be inconsistent. So let's move on. Here they say comment on these results. Here I said, people with Parkinson's disease caused by LRRK2 and those with unknown causes of Parkinson's have lower mean numbers and lower mean length of dendrites compared to people without Parkinson's disease. The mean numbers of dendrites are the same for both groups with Parkinson's disease. People with Parkinson's disease caused by LRRK2 have longer neurons, I would say, or dendrites, than the group with unknown causes. And finally, for both groups with Parkinson's disease, the error bars overlap, so these results are not statistically different. So they seem like they are the same, and therefore they could be misleading. So this brings us to the end of question 7. Let's continue to question 8. Question 8 begins with the scientific articles, so I'm going to show you. I will not read. I'll just be silent as I show you. You can go through the scientific articles slowly so that we can answer the questions later on. So slowly here, going down on. This is the first page, taking you to the next page. So here they say the scientific document you have studied is adapted from an article in Education in Chemistry, Developing Vaccines, Self-Defense Classes for Our Immune System. They say use the information from the scientific document and your own knowledge to answer the following questions. Part A says Ebola virus can infect endothelial cells. Describe how this virus can cause internal bleeding. Endothelial cells are within the blood vessels, like blood capillaries. If the Ebola virus infects those blood capillary cells, it means they are going to go through the lytic phase, and when that happens, the cells are going to be destroyed. Destruction of the endothelial cells will cause the leakage of blood from the inside of the blood capillary into the surrounding tissues, and that can cause internal bleeding. So I said, the virus can enter the endothelial cells of the blood capillaries. Then the virus replicates within the endothelial cells. This causes lysis of the infected cell, and this forms gaps in the blood capillary walls. Then bleeding occurs due to loss of blood from the capillary. The next part says, explain how our immune systems learn to make another type of protein called an antibody. Here we have to refer to paragraph 4. The question is for marks. The space given is not that big, so you do not have to go through the whole bigger version of the immune response. So I chose the shorter version. I said, macrophages engulf pathogens, and the antigens are sorted and attached onto the MHC protein. Antigen presentation occurs. A helper T cell binds the antigen-presenting macrophage, so it will be stimulated and it will divide to produce the activated helper T cell and the T memory cell. The activated helper T cell releases cytokines to an antigen-presenting B cell. The B cell is activated to produce B effector cells and B memory cells. Then finally, I say the B effector cells differentiate to form the B plasma cells, and these will produce antibodies. So let's move on. Here they say one method of slowing the degradation of vaccine protein is to dry a mixture of sugar and vaccine on a filter. Explain why flushing with water releases the vaccine from the filter. They want you to refer to paragraph 8 and paragraph 9. And again, the key thing is we need to know the vaccine is a protein. I said when sugar is mixed with the vaccine, sugar bonds to the vaccine by hydrogen bonds. Flushing with water causes hydrogen bond formation between the sugar and the water molecules. So this means the sugar will detach off from the vaccine, and so the vaccine will be released. Part D says, during the production of some vaccines, the virus is allowed to replicate in eggs. This is from paragraph 13. Explain how vaccine produced in this way may become less effective. Every time something is allowed to replicate, a mutation could occur. So I said, there is a chance of a mutation to occur every time the virus replicates. If more mutations occur, there is a high chance that the amino acid sequence of the viral protein can be altered, creating different antigens. Since the immune response is specific, change in the viral antigen would make the vaccine ineffective because antibodies against the Ebola virus won't be produced. Let's move on. Here they say scientists are using genetically modified bacteria and insect cells to produce the antigens for Ebola vaccine. Again, paragraph 13. Describe how insect cells could be genetically modified to produce the antigen for an Ebola vaccine. We need to identify the gene coding for the Ebola antigen, and then we will insert the gene into a specific vector. This vector could be a liposome or it could be a viral vector. And then we could use microinjection to insert the vector into the insect cell. Once the genes are inside the insect, transcription of those genes into messenger RNA and translation of the messenger RNA into the protein will occur, and the protein produced will be the antigen. Here they say suggest 3 benefits of using an insect cell to produce the Ebola virus antigen. So I said, these genetically modified insect cells will be safer than the Ebola virus. For antigen production to occur, some organelles like ribosomes, the Golgi apparatus, the rough endoplasmic reticulum are required, and these are present in the eukaryotic insect. Insects would have lower mutation rates, and the antigen would not be easily changed, making the vaccine usable for long. If this was carried out in a bacteria, because they go through cell division rapidly, every time cell division occurs, there is DNA replication, there is a chance of mutation. But if you compare with insects that have a lower reproduction rate in comparison to the bacteria, there are fewer chances of having mutations. Lastly, they say suggests why the stems of the different versions of hemagglutinin molecules are the same, but the heads can be different. They are referring to paragraph 15. I said, heads and the stems are coded for by different regions of DNA. All stems in the different versions of hemagglutinin molecules are the same because they are coded for by the same region of DNA. The heads can be different because different regions of DNA code for each head per time. You could also say that those differences could be caused due to mutations, or the heads could be coded for by the same gene but by different combinations of exons after post-transcription modification by spliceosomes. And again, remember, after messenger RNA is produced, in transcription, there is post-transcription modification that can use the same gene to produce different matured messenger RNAs that are going to produce different proteins when everything came from the same gene. So there's a possibility of that. So this brings us to the end of question 8, as well as the end to this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.